In video number 4 of our protein structure and function series, what we want to talk about would be how cells are able to regulate the activity of their proteins, including particularly their enzymes. We also want to look at some techniques that scientists use in order to investigate proteins. Now, there are many different ways that cells can inhibit enzyme activity or modify or regulate it in some way. As a matter of fact, many pharmaceutical drugs in use today have been designed to interfere with the functioning of some enzyme in order to counteract a disease's effects. For example, the common cholesterol-lowering drugs are called statins. What statins do is block an enzyme that's involved in the synthesis of cholesterol in the liver. Certain cancer treatments, like for example methotrexate, blocks an enzyme that's necessary for the synthesis of DNA for the rapid replication that is a characteristic of cancer cells. ACE inhibitors are used to lower blood pressure, and their job is to block an enzyme, in this case one that produces a signaling molecule called angiotensin, whose effect is to constrict blood vessels to raise blood pressure. So the ACE inhibitors prevent the production of this enzyme. Some proteins that are active in the body or in cells carry tightly bound small molecules. We call these small molecules prosthetic groups, and those molecules add function to the protein. Like for example, on the right, what we see is a heme group. A heme group carries oxygen on the iron center. One heme binds to each globin chain of a hemoglobin molecule, which we know from the previous video that is in the form of a tetramer. Retinal is another small molecule. In this case, it binds to the protein rhodopsin, which is present in the photoreceptors. Retinal actually responds to light energy, and so it initiates the photoresponse that eventually will be used to give us vision. Inside a cell, protein and enzyme activity are constantly being modified. This modification allows the coordinated activity of all the thousands of enzymes that are acting in a cell at any one given time, so that they are not counteracting one another, and so that the optimum functioning of the cell is maintained under the ever-changing conditions within the cell. For example, as the type and number of available nutrients change, modification of enzyme activity is necessary. As temperature goes up and down, as the person or organism's activity level varies, all of these things will require changes in the activity of certain enzymes that maintain functioning. And over the long period of times during growth and development, different proteins and different enzymes should be active at different stages. If a cell were to build up stockpiles of a product unnecessarily, it would be wasting resources as well as energy. And so a cell has many ways to prevent excessive buildup of product. The most common way is by feedback inhibition, which is illustrated in this picture. In this case, a high concentration of the product, Z, serves as a signal to block the production pathway at its very beginning. Now, the metabolic pathways going on in a cell are usually interconnected, and so their feedback systems are very complicated, so that different pathways may block one another in a variety of different ways. So, how can we manage to do such control? One common way is for the structure of an enzyme to have not only its active site, but to have a second binding site that we would call its regulatory site. The regulatory site is complementary to a molecule that we would call an allosteric regulator. In this illustration, the allosteric regulator is CTP. When the CTP molecule is bound to the enzyme changes shape overall so that its active site is changed in shape as well. The bound CTP molecule turns the enzyme off. In the absence of CTP, the enzyme is active. A positive regulator that is common in many of the different reactions of cellular respiration would be ADP. ADP, as you know, builds up from the breakdown of ATP. So if ADP levels rise in a cell, ADP will act as a positive regulator to promote the production of more ATP in the cell. Other regulatory enzymes may have the jobs of adding or removing functional groups from proteins. In so doing, they'll modify the charge characteristics as well as the folding characteristic of the enzyme to which they're bound. Protein kinases and protein phosphatases are two related kinds of regulatory enzymes. They add and remove phosphate groups to and from their target enzymes. Protein kinases use the energy from ATP hydrolysis to add the phosphate group to the target. Phosphatases remove it. So they work in pairs, as you can see in the illustration. The localized charge change by the addition of the negative phosphate group will modify the target protein shape. This modification in shape affects its functioning. The target enzyme may be switched on by phosphorylation or it may be switched off. In either cases, the opposite action of the kinases and phosphatases act as an on-off switch. As mentioned, kinases and phosphatases work in opposition to one another, and the addition of the phosphate group can either be an on switch or an off switch, and the removal would be the opposite. This illustration shows a motor protein, such as myosin, undergoing a series of conformational changes that results in movement. Generally, these conformational changes occurring in a multi-step pattern will be powered by ATP binding, ATP hydrolysis, and release of the ADP and inorganic phosphate, or a similar reaction with GTP being hydrolyzed to GDP. Some examples of motor protein in addition to myosin are the polymerases for DNA and RNA, another motor protein called kinesin, and then even another kind called dynin. This illustration shows the series with the ATP binding, ATP hydrolysis, and release of the product. This would occur in a cyclic manner as a new ATP molecule binds once the old products are released. How did scientists figure out these properties of proteins? So the question is, how would we, or any scientist, study a protein? Well, the first thing that has to happen is that the protein needs to be isolated. In order to obtain the protein, we would break the cells open by a process of homogenation. The homogenation that is illustrated in this figure would be simply using a laboratory homogenizer, which looks similar to a blender. There are other instruments that you'll see in lab that also can be used for this process. Once the cell's contents have been released, then we would use centrifugation to separate the contents. The spinning in the centrifuge would have a liquid portion that's called a supernatant, and it produces a pellet that settles to the bottom. The heavier materials are in the pellet. Based on the G-force at which the centrifuge is operating, the components of the pellet would vary. Because proteins are so very diverse, we use their differences in size, shape, charge, hydrophobicity, and their affinity for other molecules as a means whereby we can separate them from one another. This will allow us to study a protein in isolation. Techniques have been developed that allow you to separate your protein of interest from others that would be in the homogenate by using its affinity for certain binding partners, by using size exclusion, whereby your protein is separated from others that are smaller or from others that are larger. You could also use charge attractions or repulsion as a means of separating your protein from others. And finally, you could also separate it by its solubility under certain pH conditions or under certain concentrations of salt or other solutes. Some of the techniques are by name would be column chromatography, salting out, electrophoresis, dialysis, and there are many other possible ways of exploiting these properties as well. In your textbook, for more information, you may want to look at panels 4-3 and 4-4 at the end of the chapter. Here at LR, we have the equipment we would need to use any of the previous techniques described on the slide. This particular slide describes mass spectrometry. We have a mass spec at LR as well. Mass spec would be used to help determine a protein's amino acid sequence or to determine phosphorylation or other modifications of particular amino acids. X-ray crystallography and NMR, or nuclear magnetic resonance, also are ways to be able to study a protein, in this way studying its three-dimensional structure. This illustration indicates how X-ray diffraction patterns can be obtained from a protein that has been crystallized. When we want to analyze the properties of a new protein of interest, it helps us very much to know its amino acid sequence and to know its general structure. Proteins have structural relatedness that groups them into families, and by studying these related families, we may find a great deal of information that would be useful in analyzing this new protein.